Hello everyone. Today we are going to introduce gene mutations in stool from gastric and colorectal neoplasia patients by next-generation sequencing. This paper is published in World Journal of Gastroenterology in 2017 by USEP. This is our content. First, we will introduce why we choose this article and its research question. Next, we'll talk about the material and method to obtain target DNA and how to analyze the DNA by NGS. Then we'll discuss the result and conclusion of the experiment. And then last, we'll share our question after reading this article. This is the Global Cancer Statistics table from WHO. We can find that the third and the sixth ranks of cancer incidence and the second and fourth ranks of mortality are all related to gastrointestinal tract malignancies, 就是胃肠道恶性肿瘤. Therefore, we want to know that what is the cause of this phenomenon and how to alleviate it. After reading the paper, we found that gastric and colorectal tumors are difficult to detect in their early stages, and it's due to the lack of symptoms. Even when symptoms are present, they tend to be mild and non-specific, which may delay patients for endoscopic examination. So how can we solve these problems? First, non-invasive detection is necessary. Generally speaking, the diagnosis of GIT cancer requires a more complicated process, which is endoscopy. Non-invasive detection can reduce the difficulty of detection and help early diagnosis. Second, we know that there are many different types of microorganisms in the human intestine, which means that the stool also includes non-human DNA. Therefore, sensitive test is needed to detect the target DNA, and NGS is a good choice. Third, there have been an example previously that NGS has successfully applied for investigating mutations in stool DNA obtained from patients with sporadic colorectal cancer. Based on the above, the researchers applied the NGS method to determine whether cancer mutations could also be detected in stool samples from patients with other GIT tumors. The researchers found 87 patients, including 41 stomach neoplasia patients and 46 colorectal lesions, and 14 healthy individuals as control group. The table on the right side is the summary of stool samples collected and analyzed. After finding the subjects, the next step is to collect stool and extract their DNA. The researcher used two kits, AR spin stool DNA plus kit and dsDNA BRAZ kit. First, they mix 1 spoon of stool specimen with 8 millimeters of stool DNA stabilizer and store at -26 degrees. Next, 1.4 millimeter of stool specimens was transferred to a tube to be extracted. Third, the extracted DNA was eluted in 50 microliters of elution buffer. And the last step is using fluorometer to quantify DNA. After finishing those steps, NGS analysis of DNA can start.
moving on to the NGS part, in this paper, 20 nanogram of stool DNA is used to prepare amplicon libraries using iron ampli-seq library kit 2, and two panels are used. The first one is iron ampli-seq cancer hotspot version 2. It consists of a primary pool for 207 amplicons from an average of 2,800 hotspots and 50 oncogenes. The second one is iron ampli-seq colon and lung cancer panel version 2. It consists of a primer pool for 92 amplicons from 504 hotspots in 22 genes frequently mutated in CRC. The genes included such as Notch, AKT, PIK, JAK, ERB, MAT2K, and these are for JK are only included in the first panel. All patients with gastric neoplasia and 19 patients with colorectal neoplasia were assayed using the first one, while the remaining specimens were studied by the second one. The major reason for this is that the iron implicit cancer hotspot panel version 1 contains more genes that are commonly mutated in gastric neoplasia. But what is iron implicit? The iron implicit is an advanced tool designed for efficient and accurate preparation of sequencing-ready libraries for small amounts, usually from 1 to 100 nanogram of DNA or RNA. This kit liberates the power of ultra-high multiplex PCR to enable comprehensive genomic analysis, making it an essential component for researchers in the fields such as genetics, oncology, and molecular biology. The panel consists of a pool of oligonucleotide primer pairs, making it able to multiplex up to 24,000 primer pairs in a single PCR reaction, meaning that one can target from just a few to hundreds of genes in one run. Following simple PCR, the remaining primer are digested and a library with the remaining amplicon is prepared for sequencing. The implicit technology can accommodate two different approaches. The first one is called the gene design. This gene design approach relies on the tilling of overlapping amplicons to study the continuous sequence of interest. Overlapping primer pair must be separated into independent multiplexed PCR reactions to achieve full coverage. As a result, this approach will require two separate PCR multiplexed reaction per sample to achieve full coverage. In contrast, the second one is called mutation hotspot design. This typically results in non-overlapping amplicons which primers can be accommodated into a single multiplexed reaction. After preparing the amplicon, purify is done by using AgentCore AMP Pure XPP, this one. And the concentration of the purified library was measured on the qubit fluorometer. It detects fluorescent dyes specifically bound to the target molecule, therefore is able to distinguish between double-stranded and the single-strand nucleic acid, and it is used for further sample quantification. And here's a detailed introduction of the agent called AM Pure XP beads. It is a magnetic beads. The principle of the beads is based on solid phase reversible immobilization. And as you can see in the workflow, under certain conditions, the nucleic acid is selectively bound to the magnetic beads, while the pollutants remains in the solution. After applying magnetic fields, the magnetic beads binding with the target molecules are separated from the solution, and then the beads are cleaned to further remove pollutants. Because the combination of magnetic beads and the nucleic acid molecules is reversible, the nucleic acid can be eluted from the magnetic beads with the low salt buffer. The severe on the slides here shows how the magnetic beads is modified with hydroxyl or carboxyl groups. Their workflow is shown on the below, and the purification buffer system contains PEG and high thought ions, and the DNA is bound with beads by forming an ion bridge of DNA salt ion carboxyl group after adding the beads. The ion bridge is removed in the TE buffer without PEG and thought ions. And finally, the DNA is purified after washing and elution. Different magnetic beads, inputs, and purification buffer will bind different fragment size. Um, the purified library was diluted, and the template was prepared and enriched using ion one-touch system here, which is an emulsion PCR, as you can see in the channel A. The enrichment step is crucial for increasing the efficiency and accuracy of sequencing. After the loaded on ion sphere particles, abbreviated as ISP, we need to ensure that only ISPs with attached DNA templates proceeded to sequencing. This separates template positive ISP from the empty ones. This is achieved through a magnetic base process that selectively binds to an enriched ISP containing DNA, similar to the XPP I just previously introduced. By increasing the proportion of template-positive ISP, the system significantly enhanced the accuracy and reliability of the sequencing results. In essence, the enrichment steps using the ion one-touch system to ensure that our sequencing data is of the highest quality by minimizing background noise and focusing on the DNA of interest. This automated and efficient enrichment process not only saves time but also reduces manual handling, thereby minimizing potential errors. So finally, the sequencing was carried out in the Ion Personal Genome Machine System, abbreviated as INPGM. The sequencing steps in the INPGM system is where the actual reading of DNA sequences take place. This is a crucial phase, and the INPGM uses semiconductor sequencing technology to achieve this. Once the DNA libraries have been prepared and enriched, they are loaded onto the INPGM sequencing chip. Each well on the chip contains an ISP with a single DNA molecule attached. During sequencing, the PGM system floods the chips with one of the four nucleotides, A, T, C, or G. When a nucleotide is incorporated into the growing DNA strain, a hydrogen ion is released. This release causes a change in pH, which the semiconductor sensor will detect. Then the INPGM system translates this pH change into digital signals, which are then proceeding to determine the DNA sequencing. This real-time detection method allows for rapid sequencing, providing results in just a few hours. This method offers several advantages. It eliminates the need for fluorescent labels, reducing costs and increasing speed, making it a versatile tool for a wide range of applications.
In the study, sequencing success were 78% for stomach neoplasia and 87% for colorectal neoplasia. Here identified 25 hotspot mutations, with 20 in colorectal and 5 in stomach neoplasia, plus 9 novel mutations. In stomach neoplasms, 5 hotspot mutations were detected in APC, CDKN2A, and EGFR genes, and 7 novel mutations in genes including APC and CDH1. For colorectal tumors, 20 hotspot mutations were found in genes like AKT1, APC, and TP53, with APC being the most frequently mutated. In the table to the right lists some cosmic and novel mutations detected by ion-tolerant sequencing, as mentioned in the paper. The study successfully analyzed DNA extracted from fecal samples of patients with colorectal cancer using NGS. Moreover, the study was extended beyond colorectal cancer, demonstrating the feasibility of detecting mutations in fecal specimens from patients with gastric tumors and even benign colorectal tumors.
For colorectal tumors, the success rate of NGS analysis was 87%. The most common mutated genes including APC, TP53, and KRAS. Notably, mutations in the APC gene were frequently observed, which underscores its importance in colorectal cancer progression. In gastric tumors, they made the first in-depth study in this area using fecal samples. Among them, APC mutation was the most common change, and new mutations in genes such as NRAS, DDR2, and PTEN were also identified. So in our conclusion, the study demonstrates the successful use of NGS technology in the detection of fecal DNA mutations in patients with gastrointestinal tumors. These included patients with colorectal and gastric cancers, highlighting the broad utility of NGS-based mutation screening. Traditionally, NGS has been limited by low DNA content, but this study demonstrates the high sensitivity of NGS, increasing the diagnostic level of fecal DNA testing. In addition, the results highlight the potential for early detection of GI tumors using NGS-based fecal DNA testing. And there are some questions that we would like to raise here. The first question is, in addition to the applications mentioned in this paper, can this NGS-based method be applied to detect other types of cancer or disease? And the second question is, how does the cost-effectiveness of using NGS-based fecal DNA testing compare to other diagnostic methods? The last question is, what are the challenges and considerations for implementing NGS-based fecal DNA testing in a clinical setting? And here's our division. Thanks for listening.